Now that we know how olfactory receptors work, let's consider how the olfactory system as a whole allows us to perceive smells. The key lies in our human olfactory apparatus. Unlike our visual or auditory systems, smell hijacks an organ with another purpose, our nose. The primary purpose of the nose is not to smell, but to filter, warm, and humidify the air we breathe before it enters our lungs. But our nose also does a pretty good job at smelling. Against conventional wisdom, humans are actually pretty good at tracking the trail of a scent. Not as good as the bloodhound, but we can do the job, particularly in an open field where few distracting odors are present. Like the pinnae of the ears, the two nostrils of at least some species sample different regions of space, and the information gained from both nostrils can be used to track a scent. On the outside, human noses come in a large variety of shapes and sizes. But on the inside, the biological machinery that facilitates olfactory physiology is nearly always the same. When air and odorants enter our nose through the nostrils, they pass the olfactory cleft, a space at the back of our nose where the main olfactory epithelium is located. The olfactory epithelium is specialized epithelial tissue whose primary function is to detect odorants. It measures about 3 square centimeters on each side and lies about 7 centimeters above and behind the nostrils. In many ways, this olfactory epithelium can be considered the retina of the nose. It consists of 3 primary cell types: the supporting cells, the basal cells, and the olfactory sensory neurons. Supporting cells primarily provide metabolic and physical support for the olfactory neurons, while basal cells are the precursor cells to the olfactory sensory neurons and thus present a pool of renewable neurons in a stem cell-like state. Olfactory sensory neurons, or OSNs, are small bipolar neurons located beneath a watery mucus layer, and they're the main cell type in the olfactory epithelium. OSNs congregate to form the olfactory nerve at the basal end, but are covered with a non-motile cilia, which are hair-like protrusions at the apical end. The plasma membrane of these non-motile cilia contains the odorant-binding proteins that act as olfactory receptors, as we talked about before, which initiate olfactory signal transduction. Unlike the visual and auditory systems, which are extremely sensitive to even the smallest stimuli, it typically takes at least 7 or 8 odorant molecules binding to a receptor to initiate the action potential in the OSN. Even when initiated, olfactory sensory neuron responses are orders of magnitude slower than the visual or auditory systems. Peak currents are often not reached until 1 to 2 seconds after the odorant reaches the receptors, and it takes many seconds to reset the cell back to baseline. Axons from the olfactory sensory neurons have to pass through the cribriform plate, a bony structure with tiny holes at the level of our eyebrows, separating the nose from the brain. The axons of the OSNs bundle together after passing through the cribriform plate and form the olfactory nerve, also known as the first cranial nerve, or CN1. Interestingly, the olfactory nerve is one of the central nervous system's nerves capable of regeneration, making it a useful model for neuronal regeneration studies. From the olfactory nerve, olfactory information is passed on through the mitral cell projections in the olfactory bulb, a blueberry-sized extension of our brain just above the nose level, which serves as the first critical relay station where olfactory information is processed. There are actually 2 olfactory bulbs, one in each brain hemisphere, receiving incoming inputs from the left and right nostrils. Sensory neuron axons synapse onto mitral cells in the olfactory bulb, forming spherical glomeruli that contain the incoming axons of the OSNs. Typically, each OSN converges onto 2 glomeruli, one medial and one lateral. Under certain conditions, such as head trauma or sinus illness, axonal projections from the olfactory sensory neurons to the mitral neurons can be destroyed. For example, a hard blow to the front of the head can cause the cribriform plate to transiently move or even fracture, severing the axons from the OSNs to the mitral cells. As a result, one may lose the total ability to smell, a condition known as anosmia. Anosmia is also a symptom of normal aging process, much like sensorineural hearing loss, and it's estimated by the age of 85, about 1/2 of the population is effectively anosmic. Curiously, anosmia often also causes a profound loss of taste, in addition to the loss of smell, highlighting the functional and neurophysiological connection between these two senses. From the mitral cells in the olfactory bulb, olfactory information is passed on to the primary olfactory cortex and other brain structures, most notably the limbic system. The primary olfactory cortex is the cortical area where incoming information is first processed. Olfactory information also travels to the amygdala hippocampal complex, as well as the entorhinal cortex and the medial temporal lobe. These areas are part of the limbic system, a complex set of subcortical brain structures. The limbic system isn't a separate functional system per se, but rather a collection of functional units involved in many aspects of emotion, memory, behavior, and motivation. Unlike the other sensory systems, olfaction is quite unique in that it directly innervates the limbic system. This direct connection might help explain why certain scents tend to have very strong emotional associations. Despite these strong emotional associations, however, attaching a verbal label to a smell is often not so easy. Many of you might have encountered the tip of the nose phenomenon, where you encounter a familiar scent, but you're unable to put a name to it. Indeed, anthropologists have found that there are fewer words to describe the human experience of smell compared to other sensations. Theories are now being put forward to explain this disconnect between smell and language. Anatomically, olfactory information isn't integrated into the thalamus prior to processing in the cerebral cortex, whereas other classic senses, like vision and audition, are. Lastly, the majority of all olfactory processing is thought to occur in the right side of the brain, while most language processing occurs in the left side of the brain. While none of these theories can fully explain the disconnect, the next time you can't put words to a smell, don't despair. You're not alone.